Okay, it's 12 o'clock, so welcome to another installment of Polish NMR seminar series. Today our speaker will be Dr. Agnieszka Skorupa from Maria Skłodowska-Curie National Research Institute of Oncology, Gliwice branch. But before we go with the talk, traditionally I would like to remind students about the Best Student competition. So if you are a student and want to win a nice set of Polish Chemical Society gadgets, please ask clever questions to our speaker. And now I would like to introduce Dr. Agnieszka Skorupa. Her talk will be titled NMR-based metabolomics in the studies of gynecological cancer. Thank you very much for joining us. I'm stop sharing and the screen is yours.
Thank you very much for the introduction. I will share the screen.
Yes, we can see the slides.
Okay, so I would like to present some of our applications of NMR-based metabolomics in the studies of gynecological cancers. Metabolomics is the large-scale study of low molecular weight metabolites, sugars, lipids, amino acids that participate in the biochemical pathways. It serves as a downstream readout of gene and protein expression, and it provides a crucial insight into the disease mechanisms and into the phenotypes. While genomics tells you what could happen, metabolomics tells you what is actually happening. Two core technologies are used in metabolomic studies. This is mass spectrometry and NMR spectroscopy. Mass spectrometry is more sensitive, but NMR spectroscopy is more reproducible and non-destructive. In our institute, in the Department of Medical Physics, we use 400 megahertz NMR spectrometer, which is equipped in a solution state probe, which we use for the studies of biofluids and cellular extracts. We also use a magic angle spinning probe for the analysis of intact cells and intact tissues, and MR microimaging to acquire images of small animals and small phantoms. During this talk, I would like to focus on endometrial cancer and on ovarian cancer. Endometrial cancer is the sixth most common cancer among women worldwide, and ovarian cancer is the seventh most common cancer. As far as endometrial cancer is concerned, the increasing incidence of this cancer is related to the increasing rate of obesity, metabolic syndrome, and hormonal factors. Fortunately, about 70% of cases are diagnosed at stage 1 of the disease due to symptomatic uterine bleeding, which usually makes patients visit a doctor. So this leads to a high 5-year survival rate exceeding 90%. One of the most important prognostic factor in endometrial cancer is histological grade. Usually the high-grade tumors require more aggressive, more intensive treatment than the low-grade ones. But nowadays, in addition to histopathological grading, molecular genetic classification plays an important, increasingly important role in treatment planning in endometrial cancer. Several years ago, when molecular genetic testing was not routinely used in clinical practice in endometrial cancer, we aim to correlate NMR metabolic profiles of endometrial cancer tissues with histological grade. Our studied group consisted of 64 patients with endometrial cancer and 10 control patients. The patients with grade 1 tumors were diagnosed at a late stage, where the tumor was confined to the uterine corpus. The patients with grade 2 tumors were diagnosed at both stage 1, stage 2, where the tumor invades the cervical stroma but does not extend beyond the uterus, and in stage 3, where local and regional disease spread was observed. And the patients with grade 3 tumors were diagnosed at stage 2 and stage 3. In this work, we analyzed the tissue samples which were collected in the surgery room after the removal of the uterus. And the samples were kept frozen until NMR analysis. Before NMR analysis, the samples were cut to fit the disposable inserts. The inserts were transferred to the rotors, and we measured CPMG spectra using magic angle spinning probe at the spinning rate of 4 kilohertz and at a temperature of 4 degrees. This spectra provides information about the levels of amino acids such as valine, isoleucine, leucine, lactate, alanine, glutamine, glutamate, succinate, creatine, choline-containing compounds, glycine, myoinositol, and other metabolites. In metabolomics, we usually don't look at single metabolites, but we focus on metabolic signatures. So we look at the levels of many metabolites. And for analysis, we often need dimensionality reduction techniques. And the most important question is, what is the correct direction of observation in our multivariate space? According to the principal component analysis, this direction is associated with the largest variance in the data set. The second direction is orthogonal to the first one and is associated with the next largest source of variance. And the third direction is associated with only the small amount of variance and is orthogonal to the first and to the second one. The dimensionality reduction is achieved by neglecting the not important direction. And in this example, we achieved reduction of dimensionality from 3 to 2. In principal component analysis is an unsupervised analysis, where in this technique we don't consider the membership of the class membership of our analyzed objects or spectra, but we also use supervised analysis partially square discriminant analysis or orthogonal partially square discriminant analysis. In partially square discriminant analysis, in addition to the X data matrix of our spectra, we also use Y matrix, Y space, which encodes information about the class membership or specific features of our objects. And the aim of the analysis is to correlate the covariance between both spaces, between the X spaces of spectra and between the Y space of class membership of the objects. In orthogonal partially square discriminant analysis, we also split the total variability into the predictive one and into the orthogonal one, which is not related to the problem we are trying to solve. In multivariate analysis, we usually present our results in the scores plot and in the loadings plot. Each point in the scores plot represents our objects, a spectrum. And this plot gives information about the clustering of the spectra, about the grouping, and the loadings plots gives information about the reasons of this grouping of our objects. In the loadings plot, each point is an original variable. For example, the chemical shifts of our spectra. These two plots, the scores plot and the loadings plots, are complementary. It means that, for example, the blue objects are characterized by the high level of the variables located in the same quadrants in the scores and in the loadings plots. And coming back to our endometrial cancer analysis, the scheme of the multivariate analysis we did is presented. We started from principal component analysis of the whole of our whole data set. And we found that in the scores plot of the first two principal components, the healthy tissue in black, the grade 1, stage 1 tumors and grade 2, stage 1 tumors are separated from all the remaining samples. When we look at the scores plot constructed for the first and the third principal component, we can see that the healthy tissue is separated from all the cancer tissue, and we can observe a nice separation between the most aggressive grade 3 tumors and the remaining samples. Next, we used supervised partially square discriminant analysis to differentiate different grades of the tumors. And we found that it is possible to separate these tumors quite well. And additionally, we observe within the pathomorphological grade group, we can observe a subgrouping with respect to the disease stage. So we can observe a nice trajectory. We start from grade 1, stage 1 tumors, and we go through grade 2, stage 1, grade 2, stage 2 and 3 tumors, and grade 3 tumors subdivided into the stage 2 and stage 3. We also used orthogonal partially square discriminant analysis to obtain detailed information about the metabolic differences between the control tissue and different grades of the tumor, grade 1, grade 2, and grade 3. The pathway analysis revealed that glycine and serine metabolism and phosphatidyloethanolamine biosynthesis were the pathways disturbed in all the analyzed tumor grades. Phosphatidylcholine biosynthesis and was aberrant in grade 1 tumor, beta in metabolism in grade 3 tumors only. The scores for the inositol metabolism were found to increase with grade, and the opposite was found for citric acid cycle pathway. Also were successful in differentiation between grade 1 and grade 2 tumors, grade 2 and grade 3 tumors, and grade 1 and grade 3 tumors. We used orthogonal partially square discriminant analysis to obtain these models. And I hope that in future we will be given opportunity to continue our studies of endometrial cancer, but with combination with genetic molecular classification. And now I would like to move to ovarian cancer, which is called a silent killer because the symptoms of the disease are unspecific, and the disease remains asymptomatic until it reaches late stage. So unfortunately, over 70% of cases are diagnosed at a late stage, and this significantly reduces the survival rate of the patients. Even after successful initial treatment, approximately 70 of patients experience a cancer recurrence. Ovarian cancer is a very heterogeneous disease. The most common histological type is high-grade serous ovarian carcinoma. This is also the most aggressive histological type. And according to the Cancer Genome Atlas, ovarian cancer can be high-grade serous ovarian cancer can be divided into 4 subtypes: immunoreactive, differentiated, proliferative, and mesenchymal. The immunoreactive one is associated with the best prognosis. It is associated with strong immune cell infiltration. The worst outcome is observed for the mesenchymal subtype, which is characterized by the high stromal activity and high activity of the fibrotic processes. This classification is not routinely used in clinical practice, but it shows the importance of the cancer tumor microenvironment. Not only cancer cells are important in ovarian cancer, but also the tumor microenvironment. It should be emphasized that the tumors should not be considered as clumps of bad cancer cells, but they should be considered as dysfunctional organs containing both cancer cells and the supporting stroma. The stroma contains immune cells, cancer-associated fibroblasts, vessels, extracellular matrix, and these stromal components play an active role in the growth of the tumor, in the metastasis processes, and in drug resistance. Sometimes the stroma is fibrotic. It means that it consists of connective tissue formed by excessive accumulation of extracellular matrix components. It is generally accepted that molecular heterogeneity is at least partially responsible for the high mortality rates and resistance to therapy in ovarian cancer. And in our department, we are now working on the project related to evaluation of metabolic and transcriptomic heterogeneity in ovarian cancer. And in this project, we measure NMR spectra of serum before and after the surgery. We measure NMR spectra of ascitic fluid and the NMR spectra of tissue samples. And maybe I will present some of the results obtained in this project. Our studied group consisted of 44 patients diagnosed with high-grade serous ovarian cancer, 18 patients diagnosed with benign ovarian tumors, and 14 control patients. In this work, we measured NMR magic angle spinning spectra of intact tissue samples. We acquired CPMG and J-result spectra for each sample. After NMR analysis, the tissue samples were retrieved from disposable insert, and our histopathologist evaluated the percentage content of our samples. He determined the percentage content of cancer cells, epithelial component in the benign tumors, necrosis, inflammation, fibrosis, calcification, normal ovary admixture, vessels, and fatty tissue. In 17 samples collected from tumors, we did not find any cancer cells. These samples were mainly composed of normal ovary. In a group of samples, we found that the percentage of cancer cells was from 1 to 19%. These samples also were contaminated with normal ovary tissue. In 55 samples, the cancer cells percentage, it means tumor purity, was above 20%. The median cancer cell content in these samples was 40%. And these samples also contained high content of fibrotic stroma. The samples obtained from the control group were dominated by the normal ovary tissue, and those from benign tumors were dominated with fibrotic stroma, with only a little amount of epithelial compartment. To facilitate comparison of metabolic properties of different tissue components, we selected this specific samples which fulfilled our criteria. So the samples were considered representative of cancer when the fraction of cancer cells was above 60%. The samples were representative of fibrotic stroma within malignant tumors, where when the fibrosis fraction was above 70%. The samples were representative of fibrotic stroma within benign tumors, where when the fibrosis fraction was above 80%. Normal ovary tissue was required to be above 90%, necrosis also above 90%. I am now presenting the histological images of tissue samples which were retrieved from a disposable insert. We can observe cancer compartment, fibrotic stroma within malignant tumors, fibrotic stroma within benign tumors, and normal ovary. And now I'm presenting CPMG spectra obtained for the cancer compartment, for the fibrotic stroma within malignant lesions, for the fibrotic stroma within benign lesions, and for the normal ovary. We can also, this spectra also provide information about a range of small molecular weight metabolites. And our multivariate analysis was started from inclusion of all our spectra in principal component analysis. We included the samples obtained from patients with ovarian cancer. These samples are colored according to the cancer cells percentage. We also included the samples from patients with benign tumors and from a control group. And we can observe that the samples containing above 20% of cancer cells are accumulated in the fourth quadrant of the scores plot. And these samples are characterized by the high phosphocholine, myoinositol, glycerophosphocholine, lactate, glycine, alanine. As a next step of our analysis, we included only the samples collected from patients with cancer. And we found that the first principal component is associated with cancer cell fraction, and the second principal component is associated with admixture of normal ovarian tissue. In the next analysis, we excluded the samples contaminated by normal ovarian tissue. And the scores plot obtained from this analysis shows that the samples containing cancer cells can be separated from the samples containing mainly the tumor microenvironmental components such as necrosis, fibrosis, and vessels. Finally, we focused, our analysis was focused on the samples which were found to be representative of specific tissue components. And we can observe that the samples representative of cancer are separated from the samples obtained from both normal ovary and benign tumors. Interestingly, the samples containing high percentage of cancer cells can tend to be separated from those containing only the intratumoral stroma. These samples are in green here. And interestingly, the samples representative of the stroma in the malignant tumors can be separated from the stroma collected from benign tumors. And we used supervised orthogonal partially square discriminant analysis to find a detailed interpretation of biochemical differences between the stromas in the stroma samples in the malignant and benign tumors, and we found that we found increased alanine, lysine, acetate, glutamine, glutamate in the malignant tumors. These changes were statistically significant both in univariate and multivariate analysis. Multivariate analysis also shows increased choline, taurine, and ascorbate in the benign tumors, but these changes were not visible in the univariate analysis. We performed similar analysis both for CPMG spectra and for one-dimensional projections from J-resolved spectra, from two-dimensional spectra. We also used orthogonal partially squared regression model to correlate the metabolic profiles with tumor purity. It means cancer cells percentage in the sample. And we obtained a good model for the samples containing cancer cells and fibrotic stroma. The most important metabolite correlating with tumor purity was phosphocholine and glycerophosphocholine. The cancer cells fraction predicted by our multivariate model was found to correlate well with the actual cancer cell fraction determined by the pathologist. And now I would like to present our application of NMR metabolomics in monitoring of oncolytic therapy. Myxoma virus causes a disease, myxomatosis in rabbits. This virus is not harmful to human, but it can selectively target cancer cells because cancer cells have defective antiviral pathways. The myxoma virus infects the cancer cells, replicates, and eventually causes the cell lysis. Another mechanism of action is related to the immune activation. In our institute, the group of Professor Jazowiecka-Rakus and Dr. Sochanik have extensively studied this virus. But to date, molecular metabolic properties of the infected cells have never been studied. So we aim to fill this important knowledge gap, and we used two approaches to study the metabolism of the infected cells. Solution state analysis of cellular extracts and magic angle spinning analysis of intact cultured cells. We used dual phase extraction with chloroform, methanol, and water. And we measured NMR spectra of both polar phase and lipid phase. In the analysis of intact cells, the cell suspension, the cell pellet was just transferred to the disposable insert, and we measured NMR spectra in NMR spectrometer. The scheme of the multivariate analysis is presented. We analyzed 4 cell lines. 3 of them were found to be sensitive to viral infection. The infection caused decreased viability of these cells, and 1 cell was not permissive to the viral infection. As I mentioned, we analyzed both extracts and intact cells. And now I'm presenting the noisy spectra obtained for both control and the infected cells in the aliphatic region and the aromatic region. We can observe the signals of amino acid, lactate, alanine, uridine, nucleotide sugars, glutamine, glutamate, choline compounds, glutathione, and many other metabolites. We also analyzed one-dimensional projections from 2D J-resolved spectra. Principal component analysis of the noisy spectra revealed that the control and the treated cells are separated for the cell lines that were found to be sensitive to the virus. This effect was not evident for the cell line which was found to be non-permissive to the virus. And next, we used orthogonal partially square discriminant analysis to obtain detailed information about the control and the treated cells for each cell lines. In this analysis, we focused on the metabolites, on metabolic changes that were common to all cell lines sensitive to viral infection, and the changes not detected in the non-permissive cell lines. And we can notice that after virus treatment, we can observe increased glutamine in all cell lines sensitive to the myxoma virus, decreased phosphocholine, decreased succinate, increased uridine nucleotides or uridine nucleotide sugars. Pathway analysis also revealed some common biochemical pathways disturbed in the cell. In the cells sensitive to the virus, it was phosphatidylcholine biosynthesis, amino sugar metabolism, aspartate metabolism, and citric acid cycle metabolism. And interestingly, this analysis did not reveal any significantly altered pathways for the cell lines for the same cell line that was not permissive to the virus. And as I mentioned, we also studied intact cells using magic angle spinning spectroscopy. The nose spectra are presented. They are dominated by the broad lipid signals and the signals of choline-containing compounds. To facilitate detection of low molecular weight metabolites, we used CPMG sequence, and we assessed the levels of phosphocholine, glutathione, glutamine, alanine, and many other signals. Multivariate analysis of nose spectra revealed that in all cell lines sensitive to the viral infection, we observe increased phosphatidylcholine and decreased phosphocholine. Similar analysis performed for the CPMG spectra also revealed decreased phosphocholine, and additionally, the analysis of the aromatic part of the spectra also revealed increases in uridine nucleotides or uridine nucleotide sugars, and these metabolic changes were also visible in extracts. So it was very interesting for us. Concluding, I would like to say that NMR spectroscopy is a powerful, non-destructive technique, which enables, which provides a wealth of knowledge about possible biomarkers or disease mechanisms. So it is very helpful in oncology and in for analysis of gynecological cancer. I would like to acknowledge our collaborators from our Department of Medical Physics, from the Medical University of Silesia, from Cardiomed Silesia, and from Tumor Pathology Department and Center for Translational Research and Molecular Biology of Cancer. Thank you very much for your attention.
Thank you very much for a very interesting talk, and we are open for the discussion. I especially encourage students to ask questions. If you want to ask questions, please turn on your microphone and, if possible, camera. Do we have any questions? So maybe I'll start with a probably quite simple and naive question. So I just wonder, like, what are the clinical benefits of doing the NMR metabolomics of tumor compared to the traditional histopathology?
In routine clinical practice, histopathology is a basic technique, and doctors.
Like basically, from your first two projects, what information or that are important for doctors basically we're getting out of the NMR analysis here?
Several years ago, magic angle spinning NMR spectroscopy was proposed as a good intraoperative tool. Sometimes the information, medical information, should be available during surgery, and NMR is very fast, reproducible. So many groups around the world tried to use this technique for obtaining clinical information without histopathology, because it is more. It is faster. If we have multivariate models, developed multivariate models, we can just project our spectra onto these classification models, and the information about the class membership of this spectra could be obtained maybe in 20 minutes. So it was proposed, but as I mentioned, metabolic information tells us about the phenotype. So routine histopathology is based on morphology, and using metabolomics, we obtain information on a different level, on the level of metabolites and products of metabolism. Potentially, it is the source of both prognostic, diagnostic biomarkers. So I think it was the aim.
Okay, thank you very much. I think Krzysztof has a question.
Yes. Thank you for the nice talk. My question is as follows. So I understand that these samples, they come from how it's called, biopsy, right?
Yes.
It's a small fraction of the tumor, right?
Yes.
So did you try, or did anyone try doing 2 biopsies of the same tumor to see how similar the samples would be?
Yes, I presented information about our project related to ovarian cancer. In this project, we analyzed several samples from the same tumor, just to analyze the molecular metabolic heterogeneity. And we collect a huge database of samples of spectra. And after analysis, I think we will obtain a picture of metabolic intratumoral heterogeneity.
Oh, okay. And the second question would be about the measurement time. So what were the fastest spectra that you acquire? I guess CPMG, right? Those.
Yes.
But how long does it take to acquire one CPMG?
Our parameters enable spectra acquisition in about half an hour.
Okay. Half an hour plus sample preparation is, I guess, longer, right?
Yes, shimming, manual shimming.
No, simply I wonder if anywhere in the future would it be possible to use NMR metabolomics during surgery, to support surgery, to analyze tissues during surgery. But I guess we are very far from this, right?
There were some attempts. Theoretically, this could be possible, because I think that if we had higher amount of sample, the spectra could be acquired in 10 minutes, for example, just for acquisition. Additional time would be needed for shimming, but I think that 15 minutes should be okay. And as I mentioned, if we develop multivariate models, classifying normal and diseased tissue, we could just project our spectra onto our projection planes, and this could give information about the class membership of the spectra. But there were attempts, many groups tried to do it.
Thank you. Thank you.
Do we have any other questions? Let's give a moment maybe for someone to decide. Okay. It seems that no one is having any more questions, so I'll thank you once more, Dr. Skorupa, for her talk, and we'll announce what is coming. So in 2 weeks, we have a talk from Łukasz Jaremko from University of Texas Medical Branch, The Age of Structural Plenty, NMR-guided chemical control of functional conformational states. Note that it would be a different hour than usual. It's 3 p.m. Unfortunately, 12 a.m. here, it's 5 a.m. in Texas, so Łukasz asked me to postpone it a little bit, the talk. And we'll have also on May 13th Jan Stanek and on June 3rd Katarzyna Malec. So thank you all for joining us and see you in 2 weeks.
Thank you.
Thank you. Bye.
Thank you. Bye.
Thank you.